Hi everyone. Today we are going to talk about a biology study that relates the length of telomeres to plumage colorations in Hirundo rustica, commonly referred to as the barn swallow. This study first appeared in the Molecular Ecology Journal. Before we can get into how the experiment was conducted and the main findings of the study, we first have to understand what telomeres are. A telomere is a repeating sequence at the end of each chromosome that acts like an end cap that protects the chromosome from deteriorating or fusing with other strands. A telomere protects the chromosome like an aglet protects a shoelace from fraying. It's important to note that after every cell reproduction, telomeres shorten. When the telomere reaches a critical length, the cell stops replicating. We are currently learning about telomeres and DNA in Module 4. This study, though, attempts to link the effects of telomere length to the
The
coloration of the
scientists
barn
started
swallow's
conducting the
feathers.
experiment by tagging European barn swallows from 11 different colonies in Milan, Italy. Over the course of 3 years, the researchers located, tagged, and marked all of the adult barn swallows in the sample area to distinguish them by number, colony, sex, and age. Then, the scientists obtained blood and feather samples from every single bird tagged. After the samples were obtained and sent back to the lab for testing, scientists in the field performed weekly inspections on all of the nests in the sample area to measure seasonal clutch size and seasonal reproductive success. Back at the lab, scientists extracted the barn swallow's DNA from the cells in the blood samples. Using analytical technology such as a nanophotometer, they were able to measure the length of the telomeres. Feather colors were also analyzed to find the color of the feathers in terms of human visible light, ultraviolet color, and color saturation. After the experiments were completed, scientists utilized various statistical analyses to determine how significant their findings were.
So, overall, the study found that males with a higher relative telomere length have a darker ventral visible coloration, higher saturation levels, as well as a different UV coloration when compared to their female counterpart. All showing low p-values, this indicates a high probability of the observed traits following the hypothesis. Females didn't have a strong correlation between darker visible ventral plumages and a higher telomere length, but did have a correlation that showed that higher telomere length leads to higher UV coloration. Similarly to other bird species, females had lower ventral plumage saturation compared to their male counterparts. The relative telomere length did not differ between males and their mates. It also did not vary in age in both males and females. From the figure, you can see that seasonal clutch size was not predicted by the RTL of the female, indicated by the high p-value. Age, however, was. The seasonal fledging success, or number of eggs that produce fledglings, increased with the relative telomere length of the females and males. Seasonal reproductive success also increased between both, specifically darker males. As said before, there was no correlation for darker coloration with the relative telomere length for females. The study found that an increase of telomere length by 2 standard deviations increased seasonal reproductive success by 24% for males, 25% for females.
After performing statistical analysis on the collected data, the final results showed that there was a correlation between the barn swallows with the longer relative telomere length and their reproductive success. Furthermore, there was also a strong relationship between the swallow's plumage coloration and the relative telomere length. This suggests that the barn swallows are able to identify, indirectly, the better mate with the longest telomeres based on just the plume coloration, and will tend to choose a mate with a more desirable coloration. From a different study, it has also been shown that color darkness of mates is under directional sexual preference and coloration is involved with mate choice. So when a female barn swallow chooses to mate with a darker male, she is subconsciously choosing the male with the greater reproductive success without even realizing it. This behavior actually drives the species forward and increases the reproductive success of the colony as a whole. The genome of the barn swallow affects its physical appearance, which in turn affects their mating success. In another study by Patrick Carroll, the same relationship was also shown between tawny owl plumage and their relative telomere length, which shows that this is a behavioral and physiological trait not limited only to barn swallows. In conclusion, this study showed for the very first time in any bird species that telomere length covaries with both reproductive success and coloration in both sexes.